Hi, everyone. Welcome to the first of the webinar series. My name is Alina Mushtaq, and I'm the member of the Ensembl outreach team. And I will be presenting today's webinar on exploring genes and genomes with Ensembl. I'm just going to stop my video for now whilst I present, just so that the voice quality is better. So for this webinar, we're going to cover what Ensembl is, how to browse and navigate the Ensembl website, how to search and visualize data, customize a page, and the different types of data that is available on the browser. And then finally, and the most important one, we're also going to talk about where you can find help on and support on Ensembl. So why do we need genome browsers? Genomes are big and they contain a lot of data, and we're producing them at a fairly massive rate. And if we look at the sequencing data, it can look a bit like this. So it's a string of letters of genetic code, and this data is completely meaningless without annotation. The only way this data will mean something is if we work with it and process it, and this is what genome browsers do. So Ensembl is a type of genome browser that takes publicly available sequencing data from genomes. It's important to know that we do not perform any sequencing ourselves. So we take the genome data and then we make sense of that data. So we annotate genomes with genomic features like genes variants, enhancers, and promoters. And we also perform our own analysis on the data. Ensembl is also called added value resource. So this means that it brings together information from a wide range of other databases in a single site. So it provides easy access to all of this data. We have a lot of data in Ensembl. So genome assemblies are the primary data type that we import from publicly available databases. And then we annotate these genomes with genes. These are called gene builds, and we have gene builds for over 300 species. Then we have variation data. So not every, this is not for every species, but for a good representation of them. We also have comparative genomics, which is calculated in-house, including gene alignments, gene trees, and homologies. And we also have regulatory builds for a set of regulatory elements, and these are depicted from experimental data for human and mouse. And then we also have some tools to help you access the Ensembl data. There's a tool called Biomart, and this is an online export data tool, and this allows you to export large amount of Ensembl data without programming. We also have VEP, so VEP is short for Variant Effect Predictor, and this allows you to annotate your own variants and find out information about them. We also allow you to display your own data in the browser, and we provide programmatic access to our data via our APIs. Ensembl is open source and it's completely free to use. And you can also find our whole genome files stored in the FTP site. Ensembl is dedicated to vertebrate genomes. So there are about 300 vertebrates, and the number is constantly growing. But you can also find non-vertebrate species. So you can find genomes, fly genomes, worm, and yeast genomes. So these are model organisms. And you will find these on the Ensembl sister site, which is Ensembl Genomes. Ensembl Genomes also has Ensembl bacteria, Ensembl fungi, metazoa, plants, and protist species. And these are color-coded, as you can see on the slide, and they follow, all of them follow the same logic and organization as the main Ensembl site. Ensembl data is updated every 3 months as genomic data constantly changes and grows. So our data is not static. Every 3 months, we release a new version, and there's a lot of behind the scenes work. So we add new genome assemblies and we update our gene sets. We also update our software. We run new analysis on the data. We update our variation and regulation data, and then we finally package it all up and display it in the browser. The current Ensembl release is Ensembl 103, and for Ensembl genomes, it is 50. This will get updated sometime in May. So this means that if you come back to Ensembl in a few months' time, the data there might not be the same. So it's crucial to know which version of Ensembl you're working with. And if you wanted to make your data consistent and wanted to find the version that you were using in the past, you can do so by using our archives. And I'll show you where they are stored in a moment. We store all of our versions in our archives, and these keep frozen in time for up to 5 years. So now I'm going to jump out of my presentation and move to the demonstration part of the webinar. So I will take you through the Ensembl homepage and show you how to navigate the browser. And then we're also going to look at the different data tabs that are available in the Ensembl browser. So to go to the browser, you can simply just type www.ensembl.org. And there are a few things that I would like to point out here. Firstly, you have the blue navigation bar at the top, and this navigation bar will be present on every Ensembl page. So on the bar, we have our most popular tools. So you have the BLAST, you have the link to the web. So this is the variant effect predictor that allows you to annotate your own variants. Then we have Biomart, and this is a really cool data export tool, and I'll go through it in a bit. Then you have the downloads link. This provides you the link to our FTP sites, and you have help and documentations, and then you also have a link to our blog. So then we have 2 search bars on this page. So this is the main search bar, which allows you to search for genes. You can search for genomic locations, a variant ID, and also by phenotype. And then you can restrict these features to species. So you can select your species from this drop-down menu here. You can also use the corner search bar on the right-hand side on the top of the page. And over here, you can search by all species. So if I scroll down a bit, over here in this section, you have all the genomes that are available on Ensembl. And you can use these links to go to the genome page. You can also view the full list of all the species. So I'm going to go to the human genome page from here. And this leads me to a page where I have more information about the current genome assembly. So this is GRCH38 for humans. I can find out more information and statistics. I can also download the DNA sequence in FASTA format. And I have more examples on the gene annotation, comparative genomics, variation, and regulation data. So I'm going to go back to the homepage now by clicking on the Ensembl logo here. And if we scroll further down the page, in this section over here, we have the links to the sister sites, so all of the Ensembl genomes. So you have the Ensembl bacteria, fungi, and so on. And then on the right-hand side, you have this small link here that says View and Archive. And if I click this, this will show you a list of all the archives, and these are arranged in chronological order, except for GRCH37. So this site is dedicated to the previous human genome assembly. So I'm just going to jump out of it. And On the right-hand side, you also have information about the current release. So this is Ensembl release 103, as I mentioned, and this was released in February 2021. You then have a few bullet points on the updates in this release, but you can also find out more information about releases and updates, and also job advertisement on our blog. So we have the links to the blog here. Now I'm going to move on to showing you the different data tabs that we have in Ensembl. So I'm just going to search for BRCA2 gene in human. And this leads me to the gene tab. And the gene tab shows me information about the name of the gene. We also have the Ensembl stable ID, the description of the gene, location of this gene, and then the summary section over here will give me more information about this BRCA2 gene, and also some information from external resources. If I scroll down, I also have the gene diagram in the context of the genome. And then we also have, you can also see all the transcripts of this gene. So by clicking show transcript table here. So I'm going to go on to the transcript tab later, but I just wanted to bring your attention to the left-hand side menu over here. So you can select loads of things here. So you can select comparative genomics. You can show gene trees for your gene. So this is over here. And if you don't, if you want to find out more information about the image that you're looking at, you can also click the question mark over here. So if I just click this, it will then show me, it will give me more information about what the image is. So I'm going to go back to my gene tab. And now we're going to go to the transcript tab. So I can go to the transcript tab by clicking on one of the transcript IDs here. And this transcript tab, again, gives me information about the transcript, the location of this transcript, the gene that this transcript is a product of, and then this table, so this transcript of our choice. So all of this information is from this transcript that is also highlighted in blue here. We then have the diagram of the transcript, and then some more information like statistics, the type of transcript this is, annotation methods, and so on. So now I'm going to jump back to the gene tab over here, and I want to talk a little bit about the variation data that we have. So to find out all the variants that fall within this gene, I can simply go to this genetic variation section here and click on variants table. And this will now show me all of the variants that fall within the BRCA2 gene. This table gives me information on the variant IDs, their locations, alleles, the type of variant that they are, consequence type, and also pathogenicity prediction scores. So to go to the variant tab, I'm going to click on one of the variant IDs here. So I'm going to choose the first one. And now this variant tab gives me information about my specific variant. So I have information like most severe consequence. So this is a missense variant. I also have information on the associated alleles, location again. We get information about the different other formats of this variant. So we have VCF, and we also have HDVS notation. And then you can also explore this variant further by clicking on these links here. So we can explore it in genomic context, genes and regulation. You can also go to population genetics, where you will find more information on allele frequency. And And now I'm going to go on to the location tab. So let's go here. So what the location tab shows me is the region in detail. So this allows you to browse genes, variants, sequence conservation, and other annotation along the genome. There are three main images or panels on this page. So you have the chromosome, then you have the overview. So this is a 1 megabyte overview. And the third is loading right now. But what this will show you is region in much more detail. So we have this over here. And you have all the data as tracks. And you can also, if you want, add loads more tracks by clicking on Configure this page tool here. So if I click on this, I can select loads of other data tracks like genes and transcript, variation, regulation. So this will give me information from a regulatory build. And you can also add custom tracks or attach public track hubs. And then you can also view and edit your own personal data by going to this personal data section here. So I'm going to jump out of it. So I hope that this has given you a good overview of the different types of data that we have in Ensembl. Obviously, there is a lot more to explore. However, we will need a day or two to go through everything in Ensembl. I'm going to now look at the web tool and then the Biomarch tool. So web, you can access it by clicking on this shortcut here. And this leads you to a page where you get information about the different types of data that you can get out of web. And you can use the web in three different ways. So you have the web interface. So this is super easy to use. It's point and click, sometimes copy and paste, and this is suitable for smaller volumes of data. Then you also have the command line tool. So this option gives you more flexibility, and it's suitable for large volumes of data. And you can also use this offline. So for example, if you were working with patients' data and you wanted it to be private, then this will come in really handy. And then you have the REST API option. So this is language agnostic, and it's ideal to use as part of a pipeline, or if you want to embed your data in web pages. So let's just click on the web interface. So I'm going to click on Launch Web. And to make your web query, you will need to first select your species, and then you can input your data here. So you can paste your data over here. So this could be in different formats like genomic coordinates, VCF, it could be variant IDs from DBSNP, HUVS notation, and also SPDY. Or you can also upload your file or provide a URL if your data is stored on a public directory. Then you will need to choose the transcript database, and you can also finally use additional configurations. So these are all the different things that you want in your output. So you can have additional identifiers, you can find out information about the co-located variants and the frequency data, and you can also find out about variant predictions. So this could be SIFT and polyphen. And now I just want to explore the Biomarch tool. So this is our online data export tool, and this will come in really handy. So for example, right now we just looked at the BRCA2 gene, but if you wanted to look at a set of 100 genes and you wanted to find out their phenotype description, perhaps also gene ontology terms, and you also wanted to convert, you had the Ensembl gene IDs for these genes, but you also wanted to convert them to external IDs. So for example, the NCBI IDs. So instead of going on the Ensembl site and going from one page to another for 100 of genes, you can get all of this data in just one place by using Biomart. So to use it, you will first need to select your data set. So this could be Ensembl genes, Ensembl variation, Ensembl regulation data, and you will also need, sorry, this is the database, and then you will also need to select your data set. So this is the species that you want to work with. And then for filters, these are things that you want to search with. So for example, if I want to search with gene names, I will simply input all of my gene names here. And then I will go to attributes. And these are things that you want to output. And from here, you can select loads of data like transcript IDs for your genes, phenotype information. This will also give you gene ontology terms and also external references. This is just a quick overview of the different data and different really cool things that you can do with the Ensembl browser. I'm now going to go back to my presentation and just talk about where you can find help on Ensembl. So you can find tutorials. So these are long and short courses on Ensembl and EBI training pages. We also have video tutorials on YouTube and Youku. You can also find loads of information and frequently asked questions on Ensembl help and documentation pages. And if you want to contact us directly, or if you would like an answer to something very specific, then you can email us on helpdesk@ensembl.org. You can also join our public mailing list to keep up to date with Ensembl resources. And if you would like to host a workshop at your institution to train you, if you would like us to hold a workshop at your institution to train you or your colleagues on Ensembl, then we will be happy to do so. You can also follow us on Facebook and Twitter. And you can also check out our blog for bite-sized information about the latest releases of Ensembl and updates. If you use Ensembl, please don't forget to cite us because this lets us know who is using Ensembl and how they're using our resources. And finally, I would also like to add here that Ensembl is a team project, and I would like to acknowledge the entire team that make it possible and also our funding bodies. Thank you.
Thanks a lot, Alina. Thank you very much for today's webinar, and thank you to all our attendees who attended the webinar today and for sending your questions. So before we move on, Alina, could we go to the next slide, please? Yeah. All right. I'll also like to draw attention of our attendees that we have a new edition of new online tutorial, which is called Introductory Bioinformatics Pathway, which you can find on our EMBL-EBI training webpage. So this course is basically a collection of online tutorials, and it will it has tutorials as well as recorded webinars, and it gives you an introduction to the science of bioinformatics and also gives you an overview of different EMBL-EBI resources. So could we go to the next slide, please? Yeah. And today's webinar, as Alina mentioned in the beginning, that it's the beginning of our webinar series, which is themed with a guide to concepts in bioinformatics. So keep your eye on our upcoming webinars in this series. Thanks again. And with that, we will see if we have any more open questions. Okay, so there's a new question. Is it possible to find gene of invertebrate or plants in this Ensembl or this online show only human, zebrafish, and mouse?
Yes, it is possible to find information about genomes of non-vertebrates. And for this, you will need to go on our sister site, which is ensemblegenomes.org. I can just simply go out of it. And so you can, as I mentioned, go to our sister sites from the bottom of the page. So you can select Ensembl plants here, or you can also go to www.ensemblegenomes.org, and this will also take you to the Ensembl Genomes website, which gives you, you also have Ensembl COVID browser here, and also the other Ensembl genomes.
Is it possible to download a particular gene sequence from different organisms?
Yes, it is possible to download a particular gene sequence. So you can do this either by Biomart. So if you want a sequence for a specific location, you can download this in FASTA format. Or if you want to download the whole genome, you can do so by going to our FTP site, which you can find on here in the downloads. And if you click on FTP download over here, you will find all the different species and the different sequences.
So, all right, with that, I would like to thank Alina again, and I would like to thank Emily and Nihal for answering the questions. And thank you again. Thanks a lot, everyone.
Thank you.